When I make phylogeny videos, like the ones I did on the evolution of snakes or of spiders and so on, I usually do those, as I prefer to do, in two parts. One video explaining how that category originally came about, and then another for how they diversified within that newly emerged category. This video is part of an informal series and should be nestled between this video, talking about the evolution of mammals becoming mammals, and this video, which is about the emergence and divergence of metatherians from eutherians. I've made a few videos on the evolution of different lineages of eutherians, like I did one on afrotherians and another one for xenarthrans, and those are both sister clades of the magnorder Atlantogenata. The sister set to Atlantogenata is Boreoeutheria, which is a much bigger group, so big that I've done several videos just on different subsets. I made a playlist on the emergence of whales and a couple more for the divergence of dogs and cats, and of course several on human evolution. I'll do even more videos on Boreoeutherian groups in the future because it is an enormous collective. Metatheria is a much smaller group even when you include all the known fossil forms, and Prototheria, our topic for today, is a tiny group anymore if we're only talking about the ones that are still alive. But they're also the most primitive, with the clearest and most illustrative connection to the evolutionary past of all mammals. In a previous episode, I said that if all living mammals were divided into just two groups, then one of those two would be Theria, including both eutherians, that's placental mammals, and metatherians, marsupials. The other group, Prototheria, is represented today only by monotremes. That's just two families, Ornithorhynchidae, the Platypus family, and Tachyglossidae, the echidna family, that both evolved from a common ancestor in Australasia. And what is the evidence for that, some of you say? Well, we're about to get into that. Different lines of evidence from fossil morphology, physiology, embryology, and genetics. Starting with the fact that therians, that's all placental mammals plus all marsupials, every living mammal except monotremes, all have XY chromosomes for sex determination. Reptiles and birds have either ZW systems or they have their own form of XY that is different from that of mammals. Monotremes technically have an XY system too, but it is significantly different from that of any therian. Monotremes split from the therian lineage so early that their system is profoundly different. The platypus, for example, has 5X and 5Y chromosomes, where we have only one of each, and some of their X's are structurally similar to the Z chromosomes that are found in birds. So, prototherians are markedly distinct from therians. Prototheria is another antiquated term from the 19th century, but I'm using it today because I don't want to do a video just on the platypus and 4 species of echidna. I want to include all their fossil precursors too, earlier forms of egg-laying mammals that used to be widespread, but that don't exist anymore. We're looking at the beginning, or crown, of class Mammalia, when, as we saw in the previous episode, the most recent proto-mammals were virtually indistinguishable from actual mammals, and the same is true of side branches of almost mammals that went another way, and side branches of true mammals that didn't end up where we are. Even the first true mammals weren't entirely mammalian yet. For example, let's take a quick look at the taxonomic family Tritylodontidae. This is a group of cynodonts which are normally known for their canine teeth. That's why we therians are cynodonts as well, though this is a different group. Several of these have lost their signature canines and use their incisors instead, like the original Nosferatu from 100 years ago. Otherwise, they look a bit like if weasels were rodents that laid eggs. Except, of course, that they had uniquely distinctive cheek teeth, different from anything alive today. And sadly, while we do have some skulls and postcranial skeletons for many of these early beasties, most Mesozoic mammals are known primarily by their teeth and some associated jawbones. Other bones might be overlooked if you're out looking for fossils, but a jawbone with teeth in it is hard to miss. That said, let's look at another cynodont subset and see how frustrating it is to find just this much of an animal. This is Docodon. It's definitely more like a mammal than a reptile. It's another form of cynodont, you can tell by the canines. Otherwise, the cheek teeth are equipped for an omnivorous diet, just like you would expect of a shrew, though it's not a shrew, because the composition of the jaw means that it's not fully mammalian yet. But we can't tell much more about it yet from just this. Complete articulated fossils are especially rare, like this Agilocodon scansorius, another docodont that shows us that we're back to when everything looked like a shrew. Well, not everything. Castra cauda is a docodont too, but it shows adaptations for aquatic living like a beaver or an otter, albeit on a smaller scale at just over a foot long and weighing less than 2 pounds. It lived in the Jurassic period around 160 million years ago, at the same time as some of the largest reptiles ever on land or at sea, while these little wannabe mammals were in the rivers and streams chasing smaller fish than the dinosaurs and such would have cared about. Tritylodontids and docodonts are both commonly considered to be mammaliamorpha, but not quite mammaliaformes. And they were both diverse groups, too. Each of these listed names is a genus within the family Tritylodontidae, and it seems that there were a dozen or so genera within docodonts, too. And remember that a taxonomic family includes multiple genera, and each genus typically contains multiple species, so there's more here than it seems. Indications are that all of these were egg layers, and there is no evidence that any of them produced milk. But they were warm-blooded, albeit barely, and they were furry, at least a little bit, so you probably would have thought of them as mammals if you'd ever seen one alive. Even if none of them had external ears, because that's only on our side of the family tree, not the prototherian side. In the last episode, we said that Morganocodon from 205 million years ago was pretty close to mammals and may be a mammal. Megazostrodon from around 200 million years ago is the earliest known unambiguous mammal. It should have had 5 sacral vertebrae like us modern mammals typically do, but it only had 3. And it had a couple of other primitive cynodont features that are lost in later mammals. So there was never a first mammal that was suddenly everything that you'd expect a mammal to be. Instead, we have a long, slow series of gradations from Mammaliamorpha to Mammaliaformes to eventually mammals and then the various subsets thereof. Is Morganocodon part of the mammalian lineage right there at the base of it, or just outside of it? It's hard to tell from fossils alone. If we had their DNA, then we would know. But without an example of their genome in addition to that of the other quasi-mammalian outgroups, there's no way to be certain. So where are we on this cladogram? We are in the clade of theriomorphs, whose descendants include trigonodonts and multituberculates, which are all extinct now, as well as therians, meaning eutherians and metatherians, which combined account for every mammal alive apart from the monotremes. I will probably make another video later on just on early theriomorphs leading up to theria. Today, however, we're taking another road. We're talking about this side group. Comparing the genomes of living monotremes to extant therians and computing their divergence based on the molecular clock of averaged mutation rates, there is evidence that the lineage that led to monotremes diverged from theriomorpha between 220 and 185 million years ago. The current cladogram has that lineage as Gynotheria, which then branches into Australophinida and Schuotheriidae. Although, it might be instead that Gynotheria is not a valid clade, because it's based on combining Australophinida with what might be another outgroup. Schuotheriidae, like Tritylodontidae, retain some post-dentary bones in their jaw, meaning that if they were in the genetic lineage of mammals, then monotremes descending from them must have developed their tiny bones in their inner ear independently and coincidentally, convergently, just like our lineage did too, and that isn't likely. And one point of confusion is that there was convergence elsewhere. Schuotheriids and Australophinids share a similar tooth structure with an unrelated mammalian subgroup called Tribus-Finida, in that all 3 groups had cheek teeth, molars, and premolars that could both cut food and grind it down the same way as a mortar and pestle. Now, some thought that this shape of grinding tooth should be inherited from a common ancestor, but it's just a practically applicable function that can occur in a number of forms, and it does. Each could come about independently, and we know it did at least once because there's no clear connection to Tribus-Finida. So if we already know that it happened once, why not twice, thrice, a bunch of times? Especially since each group has their own uniquely distinct version of that structure. Because while reptiles can only open or close their jaws, we mammals can swivel ours around a bit. That ability, in concert with an omnivorous diet, produces a pretty strong selective pressure to figure out how to chew your food. Consequently, some studies place Schuotheriids closer to monotremes, while others place them closer to therians. Because remember, the best way to know if an animal is a transitional intermediate is when the experts can't tell which side of whatever line it's supposed to be on. So just to be safe, let's consider Gynotheria to be an invalid clade, and we'll treat Schuotheriidae as an outgroup of not quite mammals, being as they probably are more closely connected to docodonts. That leaves us with Australophinida, if in fact that is a legitimate taxon. Because remember, science is an investigation, not a belief. And I've read arguments from some taxonomists to the effect that Monotremata should be excluded from Australopithecina to become their own separate subset. Either way, this point in their evolution brings us into the Middle Jurassic period. And one of the subsets of Australopithecina is the family Hemispheridae, with fossil forms found in Jurassic strata of both Madagascar and Argentina. Remember that until the Middle Jurassic, the tectonic plates of South America, Africa, Madagascar, India, Antarctica, and Australia were all connected to each other such that you might be able to walk from one to the other on a supercontinent called Gondwana. But by the late Jurassic, these were all starting to break apart, so that by the Cretaceous, most of them had become separate continents as we know them today, though still very close to each other. Another subset of Australopithecina is Australopithecina. Now, these are not possums or shrews, but they look the part, superficially, externally. They also bear a strong resemblance morphologically to monotremes. Remember, these are still egg-laying mammals. Everything was back then. And these are known exclusively from Cretaceous fossils in Australia, which by then Australia was on its own, only connected to Antarctica, which had also broken off from everything else, and of course would eventually become uninhabitable in the South Pole. Remember that because it'll be relevant in a moment. Also, the earliest monotreme fossils yet found are in Australia and date to the early Cretaceous, about 130 million years ago. Many species of monotremes shared Australia with dinosaurs throughout the entire Cretaceous period, but there are as yet no therians, no modern mammals at all, not even marsupials, at least not yet, not in Australia. After the Cretaceous Paleogene extinction event wiped out most of the other egg-laying mammals, monotremes were the only survivors, and only one group of them made it. But they had the island continent pretty much to themselves, at least initially, being largely cut off from the rest of the world by then. Or you'd think. More about that in a moment. When the first marsupials eventually did arrive after the end of the Mesozoic era, but when it was still possible to get there by island hopping, those first marsupials were, again, tiny little things that looked like possums or shrews. It seems that by the time placental mammals came along, even island hopping wasn't possible for them anymore, so they might not have gotten there at all until people brought them in boats. The point is that monotremes apparently didn't face much competition from more advanced mammals, not in Australia nor in New Guinea. The problem with that is that without competition, you won't stay competitive. And while monotremes kept to themselves, living casually in isolation with reptiles, theriomorphs were still on the world stage, honing their skills, branching out to get bigger, stronger, faster, and better at everything they do. Monotremes were not driven by natural selection in the same way. They're not a diverse group anymore. Their locomotion and other bodily systems are still quite primitive and inefficient in some respects, so we should expect their fossil ancestors will be too. All 5 living species of modern monotremes have something in common. They don't have any teeth. Instead, they all have beaks. There's the duck-billed platypus, of course, plus the long-beaked and short-beaked echidnas. But we know their fossil ancestors had teeth at one time, and that there was a time before that when they had as yet no beak. That's what usually happens. The first birds and turtles and pterosaurs all had teeth and no beaks. Then birds and turtles and pterosaurs developed beaks but still had teeth in them, and they lived like that for a while until all 3 of these groups lost their teeth sometime later on. So we expect the same with monotremes. Actually, the platypus does have teeth, but only as a juvenile. This is the evolutionary principle of evo-devo, where the embryo begins to develop features that its ancient ancestors had. And we see this with whale embryos starting to develop hind leg buds. But then growth stops and the fetus reabsorbs whatever traits were then lost in their subsequent or more recent evolution. This is why the platypus loses their teeth before they mature. And just as birds still have the genes to make teeth, but those genes are disabled now, monotremes had 8 genes involved in tooth production, but they lost half of them, a trait the platypus shares with the echidnas. So there was apparently a common ancestor there, implying that echidnas inherited toothlessness from a platypus, or something very like a platypus, that was already going toothless itself. Now let's look at their fossil forms. Ideally, we should find precursors for both the platypus and the echidna, which we do, including a couple giant echidnas from the Pleistocene that are 8 or 9 times as massive as their modern cousins. Murray-glossus is the size of a wombat and is believed to be the largest egg-laying mammal that ever lived. Did they have their spines yet? Certainly, though they were shorter and fewer than those of modern echidnas. And this adaptation is not as significant as you might think. Remember that spiny hedgehogs are in the same family as hairy hedgehogs, also known as moonrats. They're exactly the same animal, except that the spiny ones have a mutation that causes some of their hairs to grow extra thick, stiff, and prickly. The same thing evidently happened with echidnas. The frustrating thing about this lineage is that in all of my phylogeny videos, I show numerous transitional forms, and we have some of that in this video too, but an important one is missing. We don't have the pre-echidna form, something intermediate between what it is and the form it once was. But of course, there's still plenty of data to go on, as you'll see. Looking into platypus evolution, there should be fossil forms that still had teeth, and there are, a few of them in fact. Obdruodon, for example, is a genus of 3 species of platypus-like monotremes in the same family as the platypus, Ornithorhynchidae. So instead of saying that it's like a platypus, practically is a platypus, but technically isn't a platypus because it's a different species, in fact it's a different genus, but it's still clearly related, like a platypus cousin, we'll just call it an ornithorhynchid because that means the same thing, close enough. These 3 species of Obdruodon lived in the Oligocene through Miocene. They clearly had beaks like a modern platypus, and they still had teeth, even as adults. And what type of teeth did they have? Tribosphenic teeth, of course, even if they evidently derived that ability independently from the other tribosphenic mammals around them. Of course, if you go back far enough, we should expect to find precursors with teeth and no beaks. Unless the acquisition of the beak becomes the diagnostic feature of the clade, as some have suggested. Because it's not just a beak like a bird or a turtle or a pterosaur has. The platypus beak is more like a radar antenna because it has extrasensory perception. The platypus beak has an array of electroreceptors. Sharks use something similar. See, just being alive generates electrical fields. Even your heartbeat produces an electrical signal that a shark can track and hone in on, even if it can't see or smell you. The platypus does the same. They hunt underwater at night when there's no light, and they often hunt things that are hiding in the silt at the bottom of the riverbeds or what have you, so that there's no way to see it or smell it. But the platypus can still find them by their electrical signature. A few aquatic animals do this because water conducts electricity so much better than the air does. It's difficult to do that in the air. However, curiously, while the platypus has some 40,000 electroreceptors in their beaks, echidnas have a couple thousand of those too, even if they live on land. Having a tiny fraction of these in an environment where they don't really work so well implies that the remaining receptors are vestigial, a remnant of aquatic ancestors. And perhaps all monotremes have this trait to some degree. The oldest known fossil monotreme from 130 million years ago is also the smallest. Again, it traces back to the generalized size and shape of a shrew, like every mammalian lineage does, except maybe for the first traces of that most diagnostic feature of monotremes. It had 5 molars, where most monotremes have 4 and sometimes only 3, except for the modern ones that don't have any at all. Otherwise, it didn't have a beak in the sense that the platypus has, where the shape of it is revealed in the bones of the skull. But I've read about other indications of a beak of some sort. According to this study, the evidence for this includes elongation of the region of the rear part of the jawbone behind the molars and development of the dental canal reflecting the presence of an enlarged mandibular nerve associated with mechanoreception or electroreception. Already, right at the very beginning or crown of that clade, the electroreceptive beak becomes the most diagnostic feature. Before they had the beak, they might have been classed as a basal member of one of their sister clades. If that, because this first one, Teonophilus trusleri, is non-tribosphenic. But once they have that beak and the superpower that goes with it, then it's a monotreme. We should also expect that since the platypus and echidna are the only surviving monotremes, that during the Cretaceous period, when the only mammals were the egg-laying type, then there should be more diversity among them back then. The fossil record shows us that too. In fact, one Australian fossil site all by itself revealed 6 different species of monotremes, all living at the same place at the same time about 100 million years ago. This is an artist's depiction, which may or may not be accurate because each of these are known primarily from fossil jaws. That can tell us whether they had teeth and maybe whether they had beaks, and of course, what the type of teeth and the shape of the jaw are all unique to monotremes. So what do monotremes look like, typically? Illustrators should stay within normal homologies, the safest guess possible. For example, the genus name Daragara is simply an Aboriginal word for platypus, meaning different species of ornithorhynchid. Just above that is Parvopalus, a more generalized monotreme that probably looked like a shrew or a possum, just like every other mammalian lineage did at one time. Above that is Steropodon. The structure of its jaw implies that it too had a beak in addition to teeth that were a bit more primitive than those of the platypus, and the beak is believed to have been pointed rather than the broad bill of a platypus. When it was first discovered, it was thought to be a platypus precursor, but is now considered to be a basal form for most of Monotremata. Here in the middle is Chalicodon ritchii. When this was discovered, it was thought that its molars looked like hot cross buns. So its name is an ancient Greek word for bread roll, probably because the other scientists wouldn't let him name it hot crust bunodon, which he tried to do anyway. That really is the informal name for this animal, because scientists are nerds. At top left is Sturtodon elizabethae, which is only known from a single tooth, one that is obviously from a monotreme, but it's bigger than that of any other monotreme tooth. So the artist took the most generalized version of a monotreme and simply drew it bigger. And finally, in the Cretaceous, there were a few generalized forms that had teeth, but no skeletally supported beaks yet. Apoleos splendens fits that description, and it's known as an echidnapus because it bears traits of both echidnas and platypus. So does that make it a transitional intermediate? Not really. The confusion there is that Apoleos lived 100 million years ago at the same time as stem ornithorhynchids, not prior to them. So it's not transitional in that way. We don't have any intermediate pre-echidnas either. If we had that, then we'd know where it was found and how old it is and what sequence of changes were in what order. Without that, we have to look for other clues. What clues? Well, according to this study, a multi-gene evaluation of the echidna-platypus divergence using both relaxed molecular clock and direct fossil calibrations reveals a recent split of 19 to 48 million years ago. That is much more recent than we thought. This genetic study eliminates Apoleos as an ancestor because that was 100 million years ago. Tachyglosses diverged from ornithorhynchids sometime in the last 50 million years. And another thing to consider is that there were already different types of ornithorhynchids around by then. Importantly, while these apparently originated in Australia around 100 million years ago, there were 2 species found later on in Argentina, of all places. Patagorhynchus pasquale from the late Cretaceous 70 million years ago, and the double extra-large Monotrematum sudamericum from the early Paleocene 61 million years ago. If both of these made it to South America, which is all the way on the other side of Gondwana from Australia, then we could reasonably expect to find fossil ornithorhynchids in Africa, India, and Antarctica too. There probably are several of them there under tons of ice, because remember, Antarctica wasn't always as cold as it is now. But to be in Argentina, that far away from the point of origin at that point in time, when Gondwana was already breaking apart, means that these animals would have had to have crossed some areas that were only intermittently connected or only traversable in certain areas. The platypus may be a fine swimmer in shallow freshwater ponds, but it is not marine. This is not an ocean-going mammal. If it ended up in a saltwater environment somehow, it would have to seek an island or find its way back to freshwater shallows. The fact that both of these ornithorhynchids predate the split from tachyglossids, the family of echidnas, and the fact of their combined distribution, along with some other data that we'll cover in a moment, led to a new hypothesis which is explained in this article. Tianophilus trusleri likely possessed an electrosensitive and or mechanosensitive bill or beak, which we suggest evolved for insectivory in seasonally dark early Cretaceous polar forests. Environmental reconstructions indicate a densely forested setting that experienced months of darkness each year and periodic snow cover during winter. Under such extreme conditions, Probe feeding with a mechanoreceptive or electroreceptive rostrum would have been highly effective in obtaining food. And of course, that same strategy works even better in temperate regions when you're in the water. So that's the beginning of Monotremata. It goes on to say that we also hypothesize that tachyglossids originated in Melanesia, perhaps on the emergent Vogelkop landmass, and then dispersed to Australia during the Pliocene-Pleistocene. What's the evidence for that? Well, they go on to say that only aquatic stem monotremes seem to have survived the end Cretaceous mass extinction, which may have in part been due to their freshwater habitats where bacterial decomposition of plant remains supported residual food chains, unlike oceanic and terrestrial photosynthesis, which were profoundly interrupted. In other words, ornithorhynchins were the only monotremes left that tachyglossids could have evolved from. But wait, there's more. Melanesia maintained a separate diversification center within the Sahel region. Subsequent faunal interchange from the late Miocene to Pleistocene coincides with the abrupt appearance of tachyglossids in the Australian fossil record, as well as the development of a distinctive rainforest-dwelling marsupial fauna that became widespread across eastern and northern Australia before extending into New Guinea. New Guinea is a tropical jungle environment where fossilization is extremely rare because of many factors. There's not even anywhere you go to look for fossils because the plant life would quickly destroy whatever was exposed. Australia, on the other hand, has lots of fossiliferous areas, so that explains why there are no fossils of proto-echidnas and why they don't show up in the fossil record until after they moved to the land down under. There is also evidence that sea levels between Australia and New Guinea were only about 1/10 as deep in the Paleocene, and they know there was a land bridge there at some point because of the faunal traffic going both ways later on. The Vogelkop Peninsula, or Bird's Head region of western New Guinea, should provide possible ancestral areas within which stem ornithorhynchids inhabiting a tropical rainforest-covered island would have had extensive areas of humid forest within which to forage. In the absence of competitors, specialization for more terrestrial lifestyle could thus have feasibly evolved. So they set all the circumstantial conditions. What else you got? Several lines of evidence indicate that tachyglossids evolved from platypus-like ancestors. Indeed, embryonic echidnas possess a marginal rostral cartilage suggesting that their characteristic beak originally developed from a platypus-like bill. This goes back to the principle of evo-devo that we mentioned before. Continuing with embryology, we can see that at a certain stage the echidna has a completely normal snout just like any other mammal, and it isn't that different from a platypus, especially when that snout is covered, at least momentarily, with the cartilage you would expect of the type of bill that a platypus has. But then when the echidna matures, then it changes into a tubular beak that can't open their mouths any more than just let their tongue out. Because when they explored the terrestrial food sources, their genome shows that they were already toothless. So all they could eat were ants and termites. And this study says the same thing, that molecules, morphology, and ecology indicate a recent amphibious ancestry for echidnas. But what is especially important here is the distribution of Tachyglossidae. As you can see, the short-beaked echidna is one of the most widely distributed animals on any one continent. There are 5 subspecies all over Australia as well as Tasmania and New Guinea, where they apparently originated. All these others are long-beaked echidnas. The western long-beaked echidnas are in blue, including the sky blue patch, where they are probably all extinct now. This tiny, pathetic dot of yellow is the entire range of Sir David Attenborough's long-beaked echidnas. And in green, we have 3 species of the eastern long-beaked echidna. As a general rule, the longer an animal has inhabited a particular area, the more diverse they're likely to be there than anywhere else they might have moved to. So the fossil record has already revealed about all that it can in this case. Having living examples of these allows us to know what the fossils can't show, like their genetics and embryology. But when we're talking about monotremes, it's their physiology that is fascinating. They have the lowest body temperature of any mammal because they're barely warm-blooded. In a sense, they're barely mammals. They lay eggs from which hatch tiny, underdeveloped fetuses that then have to hide in a mother's pouch, where they lap up milk from a patch of skin on her belly because monotremes don't have nipples. Each side of this family is weird. It's surprising to hear that the platypus is the ancestral condition for these things. It shows how much can happen in 40 million years. The male platypus has a venomous spine on its hind legs that delivers instantaneous, sustained, and excruciatingly painful stings that can lead to nausea, sweats, and muscle waste. But the male platypus has another seemingly reptilian trait in that it has a hemipenis. That's a two-headed probing appendage just like lizards and snakes have. And if you liked that Lovecraftian love story, remember that echidnas have a four-headed penis. It's like two-headed but also double-donged. Even though the females only have two vaginas. They only have two vaginas? Well, sort of. They're both connected to the same orifice, and that's not a vagina, that's a cloaca. You'd think that if you had multiple vaginas, there would be evidence of that visible from the outside, especially if your partner is packing 4 probes. But no. The one and only hole they lay their eggs through is also the same hole they poop out of. So their partner always has to tap the back door because that's the only door they have. If all that sounds sickening and weird, hey, I never said it was an intelligent design. So echidnas have a four-headed penis, although they only use 2 at a time. I think it's best to use 2 at a time, don't you? And you may not know this, but marsupials have a 2-headed penis too. It looks like a tuning fork. And there's an argument for fine-tuning, except that it's not used for, well, it's only used for forking. Eutherians like us have only 1 penis. Aw. Except for a recent news story about a guy who had 3 penises. In 2020, someone else was born with 2 penises.
He has 2. 2 what? Oh, it has 2 penises. Why is it so hard to understand? Is that a thing that can happen?
Yes, it's called aphalia. I just Googled it.
Having 2 penises would be awkward, but I can still think of one way that would work. You know, it's funny that when I thought about doing a video on prototherians, my first thought was, there's only the platypus and the echidna. That's not much to talk about. It'd be a really short video. Then I remembered that we have to talk about the evolution too. The next one I do, if I do one on theriomorphs, that will be a shorter video. Maybe. Thank you for watching this all the way through. You know, these phylogeny videos are my favorite ones to make, but they take a lot of time to research, compose, edit, and so on. And science education, sadly, doesn't get the views that drama does. So if you appreciate this type of content, please consider becoming a patron, and there are other ways to donate in the video description. And thank you very much.